Good morning, thank you very much. So I just came back from a visit to Germany to visit my family, which involves my nephews and nieces, and I never actually make these trips without catching a cold, because they obviously have a better immune system or a better microbiome than I do. So I do hope my voice does actually make it through this talk. So I was asked essentially exactly what Christine was asked, what is about evidence that changes in the characteristics of the microbiota have a physiological effect, functional outcomes. And I have to admit that I really struggled with this question quite a bit, especially also since most of the studies that I'm directly involved in are not really looking at the host to a large degree. So I'm going to talk more about our ecological studies today and our evidence for how can we actually modulate gut microbiomes through diet, and specifically probiotics and prebiotics or dietary fiber. And I will then show you some unpublished data where we are starting to look at the host at the moment to address potential effects on health. So I don't want to go too much into the basis of how the microbiome influences health, but when I was researching for this talk and I was reading the review that was written by the panel from 2012, I was actually almost a bit humbling, because it was really that the conclusions that I came up with, that we are not really able to define a healthy microbiome and that we're still struggling with getting the mechanisms, is still basically where we are today. So it is probably important to point out. And do I have a pointer here somewhere? No pointer? All right. Point out, that there is still intense rationale to actually target the microbiome, and we do know this from animal models of diseases. And we clearly know here that there is an effect of the microbiome on disease, and I do think this is very well established. We do know that germ-free animals are very different, but even animals from different facilities can be vastly different in these health outcomes, and some of them are, validated or important markers in these animal models. We also have evidence for a functional or mechanistic basis for these benefits, and even some of the microbiome traits or features that provide these benefits have been determined. And I'm not going into details here because much of this has already been mentioned. Functional mechanistic basis would be, for example, an anti-inflammatory effect, improvement of barrier function, change in gut hormones, for example. And then we have microbial traits, and we heard already quite a bit about short-chain fatty acid, but clearly this is not an exhaustive list about microbiome traits. And these entire pathways have been established in many animal models. In humans, of course, the situation is much different in that we don't really have evidence for causality, and much of the associative data that we have comes from comparing healthy and diseased individual and dysbiosis. Still, and that has also already been mentioned, is there are definitely microbiome traits that seem to really pop up repeatedly if you look into these dysbiotic microbiomes, such as a drop in diversity, a bloom of enterobacteria, say a drop in short-chain fatty acids. So providing at least the rationale, despite the absence of causative information, to actually modulate these communities by different dietary strategy. And I'm going to talk about probiotics or life biotherapeutics today, also microbial intervention, and then prebiotics and dietary fiber, and a little bit about symbiotics, but not much. And my group is specifically interested in to view the microbiome from an ecological perspective, and Curtis was pointing this out, and I like the forest analogy about that we have a highly evolved ecosystem that is, at least in healthy individuals, remarkably stable even over several years. And clearly, if we want to modulate these ecosystem, ecological and evolutionary principles have to be considered. So this is just a quick outline. I'm going to talk about how can we use also some own work about how probiotics and prebiotics and dietary fiber can be used to modulate gut ecosystem, then the evidence that has to improve health, or at least some of these microbiome traits that are associated with health. And then I'm shortly at the end, but I already answered this, the question, can we really generate a healthier microbiome with diet, and how can we actually define that? And I had these slides in just to make the importance of about the, features of this ecosystem. And I was very happy to see several of the speakers actually referring to that we still don't understand to a large degree how microbiomes are shaped. And we had already a discussion, about what really, to what degree do actually these variables, these factors explain microbiome diversity that we see in human populations. And this is really just two of these studies. They were the studies from Belgium were mentioned also, which came very much to the same conclusion. But in these two studies, they were very consistent in that they said that diet and other measurable factors do only account for around 20% of microbiome variation. Diet probably around, and Curtis also threw around the value with 5, 6, 7%. This is very, very similar in actually these studies. So where is the variation then coming from? And I was involved in a study that was published this year in eLife, where we tested the hypothesis, could it be that just ecological factors actually generate variation in gut microbiomes? And we specifically tested the hypothesis that variation could just arise through the order of colonization of actually the early gut microbiome. And we did a variety of experiments in which we put different microbiomes into mice. You can see here the donor A and donor B, and we put them in different succession. I don't have time to go through the data in detail, but what you can see is actually that the microbiome is to a large degree shaped by which microbiome comes first. And this is despite all of these experiments being done in isolators. These mice get all the same diet, they're handled by the same people, and even the species pool is controlled because they receive absolutely the same bacteria by gavage. And you can see here in this MDS plot here that the microbiomes cluster dependent on how these microbiomes are actually introduced or how these hosts actually acquire these microbiomes. Again, showing that even if you control for all, at least as good as you can for all the factors, microbiomes variation can arise through colonization order. And if you now think that throughout our life histories, we're probably associated by a lot of different stochastic elements on how the microbiome is assembled, this could explain some of the variation. And I've just put this slide back in after the discussion about the geographic allocations and the non-westernized microbiome, because this is a finding that the colonization history is something we hypothesized, but this was a finding we didn't expect, is that if we actually look for the microbiomes within cages and within and across cages and across isolators, you can actually see that all of these microbiomes cluster based on how these animals are housed. And this is not what we thought. And we were, of course, well aware that there are cage effects, but we really put exactly the same microbiomes into these mice, and we did not expect this to result in a cage effect. But it really seems that even if you had the same bacteria by gavage, and we established that we can get almost 90% of these microbes in these mice, it almost seems like these microbiomes actually wander off. And we worked with Stephen Campbell, who is a microbial ecologist, and for an ecologist, this is actually not surprising, because ecosystems are not only shaped by deterministic factors, which would be things like the host genotype, the diet that we are eating, but they are also actually shaped by stochastic effects, such as dispersal and drift. And these effects, these processes are then influenced by these priority effects that we've actually shown. And clearly, if you think, I would argue that this has major implications if you interpretate actually diversity within human population, because it is really dispersal here that shapes the microbiome to a large degree, which will be majorly different, for example, in non-Western and Western population due to the differences in sanitations, also how we actually share microbes. But it would also explain some of the biogeographic patterns, because of course individuals, or also some of the patterns amongst different human groups, because they interact more with each other and exchange more of the bacteria. It also clearly shows how difficult it is to really determine what could be a healthy microbiome, given that these ecological factors actually make the microbiome vary, and they do this even in monozygotic twins, which are actually at least almost as different as microbes. And then finally, of course, we have to take this diversity into consideration when we want to modulate the gut microbiome. So what can be done with probiotics? There is quite a bit of controversial data out there. But if you look in the literature, and if you look into, for example, some of the studies and also some of the meta-analysis, I would make the argument that at least in most studies, probiotics do not have an effect on the gut microbiome. Again, if you apply an ecological perspective, and here more specifically from invasion ecology, you actually have to think about what are the traits that this incoming probiotic organism actually has to have to be able to establish a viable population, overcome the actually factors in the host, but then also this microbe actually has to compete with the population that is in this host, and then to actually change the microbial composition, it actually also has to have a certain effect through competition or facilitation or a syntrophic relationship to actually change the microbiome. And then for this, of course, this organism has to be well adapted. And this really questions on how well adapted are actually the strains that are used in probiotic species. And we talked a lot about, there is no core microbiome, but Curtis also mentioned there is definitely a microbiome of species that is found to be dominant in humans. And if you look into these dominant species, you will actually not find the species that you at least can buy in supermarkets or that are used in yogurts. For example, Bifidobacterium animalis, which is occasionally found, is not really a core member of the microbiome. So that's really kind of questions on how adapted are probiotics really to be successful in this ecosystem. And to use Curtis's forest analogy, which I like quite a bit, for me, this is really a bit like if you would try to fly, to Costa Rica and start planting some of your room plants into the rainforest, and then expecting them first to actually be there when you come back 2 years later, and then on top of it change the highly adapted, jungle flora, which is probably not going to happen unless you really have a plant that is able to be competitive in this environment. And this is something we were very interested is that there are not many studies that use an autochthonous gut organism such as Bifidobacterium longum, and I was involved in a study where we used such a Bifidobacterium longum, and as you can see here, in a subset of individuals, around 30%, we could actually establish this strain over the duration of half a year in a human subject. Also seeing that you can, in fact, establish a probiotic long-term if you use an autochthonous strain. However, as you can also see in around 60% of individuals, the strain really just washes out. And if we analyze these microbiomes with metagenomic sequencing, and what we found is that there is an underrepresentation, first of Bifidobacterium longum in these non-persisters, which means that the same species actually excludes the invasion of the incoming species, probably through competitive exclusion, but we also showed that there is a higher amount of genes associated with dietary carbohydrates. So it really seems like there is a niche that is open in a subset of individuals in which this probiotic can come in. And this allowed us to get into the Atlantic with a probiotic that actually lasts, but it also showed that if we actually look at what this probiotic then does to the microbiome, there was actually no evidence that any changes happen. So it seems like this probiotic is just inserting itself into an open niche of these individuals. And if you look at about the clusters, these samples are actually to a large degree clustered by, again, individuals showing that there is a stable microbiome. Yeah. So I would say that most probiotics do not change the microbiome. I would argue that probiotics could potentially develop if you very specifically select for strains that actually are more competitive, and also, for example, through the production of bacteriophages or bacteriocins, would actually influence the microbiome. But despite this lack of an ecological effect, clearly probiotics have been shown to do something. There are meta-analyses that show for certain targets probiotics can actually be good. This is not a meta-analysis, but this is one of the really important papers that showed actually that a symbiotic can have an effect on sepsis, and that was published in Nature. And we also published a paper last year that showed Bifidobacterium strains can actually improve barrier function. And at least in our study, we looked at the gut microbiome, and again, we had no effect on the microbiome. So it seems like probiotic might still do something through the effects that these strains actually have, but it is potentially microbiome unrelated. So what can be done with a dietary fiber? And there was already a discussion about dietary fiber, and I have to admit I didn't take the new definition into consideration. So this is done by isolated resistant starch, which probably would not be considered a dietary fiber at the moment, but it is a non-digestible carbohydrate. And this is a study that we published in 2010, and there we tested 2 different types of resistant starch and compared it with a controlled starch. And if you look into what happens with the microbiome, the regular starch being digested had no detectable effect, while the resistant starch type 2 increased Ruminococcus promii, Eubacterium rectale, with a very low and non-significant bifidogenic effect. And this was also found in some of Harry Flynn's study with resistant starch type 3. And then finally, this resistant starch type 4 actually has a completely different effect, reducing Firmicutes, increasing Bacteroidetes, mainly through an increase in Lactobacterium dystazonis, and there was a clear bifidogenic effect and other OTUs. So you can see that even just looking at a resistant starch, but with different chemical structures, we do actually have different effects, and we have an ongoing study at the moment with just resistant starch type 4, and we could confirm that there's more overlap between these types, but even there we can actually see a specific substrate-specific effect, showing that you could potentially use a specific carbohydrate to actually select for bacteria. And then these effects are large. If you look, this is one of the subjects, and as you can see, the Bifidobacterium adolescens here is enriched from around 2 to 3% to during the resistant starch type 4 period to up to 30%. So we could make this rather minority member actually be a dominant member in this individual. After the administration stops, the levels actually go back to normal due to the resilience of the actual ecosystem. And there's also this effect is not consistent. If you look at this bubble graph here that shows the actual individual effect, you can see the large black dots actually show increases, while the white dots show decreases. You can see there are major increases in several of these taxa, but you can also see that this effect is not consistent. Yeah. So, and this also has been shown in different studies, also with resistant starches. So since I'm already running out of time, I want to show you a little bit of the data from an arabinoxylan study, and this is now a different arabinoxylan than what Kirsten just showed. It's a high molecular arabinoxylan. We gave 25 grams and 35 grams to females and to males for a duration of 6 weeks, and this was the first study in which we actually looked at the gut microbiome, but also on health effect. When you look at the gut microbiome, you can see this substantial individuality, but we could actually see a statistical significant shift in the microbiome, not with the cellulose, which is non-fermented, which you can see stays the same, but here after already 1 week, the microbiome actually shifts. And then if you look at 6 weeks again, there is no different to the week 1 sample. Interestingly, arabinoxylan decreases the diversity of the gut. And this is something that has also been shown, for example, by Li Ping Zhao's study, although he even used a complex fiber mixture based on Chinese diet, but even there is actually a decrease in diversity, which might be linked to a related factor such as colonic pH, which actually drops, but we also already discussed the importance of gut transit, which is influenced by short-chain fatty acids and potentially then also reduces short-chain fatty acids. So it might be these ecological kind of effects on the actual microbiome. Again, we have a highly individualized microbiome, and just to look at the short-chain fatty acids, and we unfortunately don't have sophisticated members to measure it in plasma, so we actually went for the fecal samples, which I know has many limitations, but we could still see a very small increase in fecal propionate. But what was more interesting is actually if we looked at the individualized responses, we could actually see that in a group of individuals there was a very kind of consistent increase over the 6 weeks, while in another group it was a consistent increase during week 1, and then we actually had a drop in propionate over 6 weeks. And this is really something that I find very something that we didn't expect and that probably has to be considered. First, it shows, that the temporal measurements are very, very important of these short-chain fatty acids, but then also that in terms of there is an individualized response, and you can see then this individualized response is actually linked to significant shifts in the OTU, but also in co-abundance clusters of the microbiome. As it seems like the individualized response to arabinoxylan does also lead in an individualized response in terms of short-chain fatty acids. Yeah. We have health data. We showed that there's a significant amount of improvement in or increase in satiety, and we could also show that there's a significant difference between cellulose and arabinoxylan in terms of markers of glucose metabolism. But, and we are looking for correlations at the moment, and I don't have this data yet, but I just would like to share one piece of data that, again, really surprised me. So this was a correlation that I did with Amanda Raymertate's group in Nebraska, and there we specifically tried to determine the role of the actual microbiome in the metabolic benefit of resistant starch. And what we could show here, as you can see, that as actually hypothesized, this resistant starch type 4 improves insulin sensitivity and with very strong shifts in the microbiome, which are very strongly correlated in the metabolic benefit. But then very much to our surprise, when we did the exact same experiments in parallel in germ-free animals, we saw virtually exactly the same effect. And if you look at the comparison, you can really see how it is almost a mirror picture between these conventionalized mice and actually the germ-free mice, suggesting that resistant starches might have an effect on metabolic benefits without influencing microbiome. And this echoes a bit what Christine just said, that effects might not necessarily be the microbiome. So with this, I would like to conclude, I would argue that we do have strategies available to modulate the composition, metabolic function, and perhaps even diversity of the gut microbiota through dietary means. Of course, much more has to be done, but these studies could be designed to target these microbiome traits as at least associated with benefits. Probiotics deliver single strains, have no effect on the microbiota, at least that's how I interpret the literature, but some of the strains have established effects, and this is very well established, including in meta-analysis. Dietary fiber can have substantial effects on gut microbiota composition. They are often leading to specific shifts, which some define them as prebiotics, which have a specific effect. And the function and production of metabolite is also increased with established physiological effects. However, we still have limited evidence that these strategies result in a healthier microbiome, and I do struggle very much with the actually, even with the attempt actually to try and define such a microbiome, simply also because of all the ecological differences that we actually see in microbiomes and how this is actually in the level of stochasticity that we see there. Definitely responses to diet are highly personalized, so a one-size-fits-all approach might actually not be optimal. And clearly we are dealing with a daunting complexity. And just, I know I'm over time, but I was also asked, to give you about short and long-term goals. I would argue the short-term goal should be that we need hypothesis-driven human trials. This is misspelled, that's the problem if you change the slide in the middle of the night being completely jet-lagged, So we need hypothesis-driven human trials that target both the microbiome and actually look at validated surrogate markers of disease. The trials should be performed using an ecological and evolutionary perspective, and they should be powered to account for inter-individualized variation and potentially the analysis of subpopulations and predictive modeling. And I would be very interested to hear what statistical tools Christine is using to potentially get closer at causality. And then the long-term goal would be establish the functional and mechanistic basis for benefits on the host in humans and identify microbiome traits that are responsible. And this is very much in agreement with what Michael Fischbach just published in Cell, where he makes a strong argument that we really still lack causative information and mechanisms to actually systematically modulate the gut microbiome. And clearly, this is only the long-term goal because I think it's the much, much harder thing to do. Ideally, we should actually do this first before we try to actually modulate gut microbiomes. But clearly, we do have, I think, enough data to at least draw a hypothesis on how the microbiome could be modulation. I think this could be tested in human trials. There are some low-hanging fruit. Actually, if you look at the healthy eating plate and you look at the diet that is suggested, you could actually almost argue, and I'm wearing my nutrition hat here, that this is to a large degree a microbiome, preservation, actually diet, establishing all kind of healthy relationship. And it might well be that this is the reason why this diet is actually healthy. But we do link, and as Justin Sonnenberg mentioned, we are missing human trials that actually look at these diets for which we have good evidence that they actually have health effect, and see if some of these effects are through the microbiome. And another low-hanging fruit, we were discussing a lot the non-westernized microbiomes, which consume a much higher level of dietary fiber, which is not only much higher than what is recommended, but it's also much higher than what we have eaten throughout all of our evolution. So I would actually argue, if we think that the microbiome is important, it should actually concern us that we are eating a diet that really doesn't support the actually metabolic activities of these microbes anymore. And I do like Justin's commentary here a lot, and the concept of that we are essentially starving our microbes. This, I would like to acknowledge my collaborators and thank you for your attention.